Hello and welcome to the session on cloud solutions. My name is Csaba Peltz, and in the next 15 to 20 minutes I'm going to be the one to introduce you to ChemAxon Synergy, our approach to research data management in the cloud. Some of you already had a chance to see a presentation on our cloud solution, maybe on the last UGM. Today, I'd like to focus on how it has evolved since its first introduction and what our next steps are in the near future development. I started the original intro presentation with the same slide, showing a network of components, a network of existing ChemAxon web applications that were integrated in order to create a cloud-based solution provided by ChemAxon as software as a service. These components include generic ones like capturing chemistry data with our small molecule registration system, but also additional ones dealing with assay data capture and data visualization, and some more specialized ones like the one dealing with compliance checking. Let's start with the basics first. So if you think about data capture in general, in a typical small molecular research workflow, it can be split into two different parts. One of them starts with the chemical synthesis that happens in the labs, and all the data that is related to those chemical entities have to be captured in a way that the structures are standardized according to the company's business rules, while we make sure that the users have the opportunity to capture all the additional data fields related to their compounds. The other part of the data capture, again, has quite a strong focus on normalization, but has to deal with all the biological assay results connected to those compounds. Besides the normalization, we also have to make sure that the assay data is properly correlated with the compound synthesized, so that in case of reporting data analysis phase, you have a chance to put together structure activity tables and all kinds of different analysis related to your assay data. We wrapped up these three components into an environment called ChemAxon Synergy, and that's provided as software as a service by ChemAxon. Under the hood, the chemistry data capture uses our compound registration product, which is an industry standard chemical registration tool that's meant to deal with checking the uniqueness of all the captured chemistry data. It has to deal with normalizing the structures according to the business rules set up by the customer. It can recognize salts whenever they are submitted along with the new structures. And it also has to deal with a number of predefined custom data fields that one might want to add to the structure capture itself. Speaking of the normalization part, ChemAxon's structure checker and standardizer modules make sure that every single representation that you can imagine, let's say in terms of stereochemistry, can be easily checked and converted to your preferred representation, so that we make sure that there are no inconsistent drawings and or duplicate entries in your database. Also, during the registration, the system makes sure to offer you similar entries from the database, similar ones to the newly submitted structures, so that you have a chance to check whether it's indeed a new stereoisomer and or if it's indeed a new tautomer form a structure, or alternatively, you have a chance to accept existing entities in the database. In case of the assay normalization, the task is somewhat different. You typically end up with a huge spreadsheet that contains measured values, measured parameters for a number of assay protocols. We need to make sure that, first of all, the entities are properly captured, and at the same time, the columns themselves that contain the measured values are properly mapped to the SOPs, to the parameters or results within those SOPs, and to the data type itself, so that you have the ability to have a consistent reporting on the data later on. In case you prefer to invest the same amount of work, essentially, upfront, instead of doing them one by one during the uploads, you can prepare Excel-based templates to upload such assay data blocks. We provide example templates that you can certainly extend, and with the use of such templates, you can easily achieve a single-click upload experience and a much more smooth assay data upload process. But what next? What to do after we have a properly normalized chemistry dataset and a properly normalized assay data collection? We have had many discussions with users during the past 1 year, what their expectations are in terms of interacting with data, interacting with such scientific data. And all these expectations essentially fell into two different categories. One of them being the easy-to-use, quick overviews of the data, an easy-to-use dashboard that presents all the new data related to a certain project, or an easy-to-use dashboard that visualizes how the selectivity of certain compounds looks like in terms of different targets. While the other set of expectations were much more related to the really flexible, in-depth analysis of all the data, which is, well, it means that that the users wanted to pick essentially any two measured results, values, and needed the ability to quickly interact with them in a way that they can find easily correlations, they can find patterns within their data set. So these are the two main tracks of data visualization needs. In order to address them, we decided to bring in a third-party tool called Tableau for data analysis. And we started integrating Tableau into the cloud solution so that we can serve the quick and visual dashboard use cases, as well as the visual data exploration. That is quite a strength of Tableau as a data analysis tool by itself. In order to make it properly embedded in the solution, besides attaching it to the data sources that we typically create, we started developing extension modules for Tableau that can deal with the chemistry part of the data. Some of them are dealing with individual data values and how those data values can be converted to a visual representation of the structures. Essentially, any kind of data field that can describe a structure, being a name, being a smiles, or any other chemistry file format, can essentially be converted on the fly to a structure representation of your choice. The same visualization has been embedded into a spreadsheet-like component in order to fulfill the typical use cases of spreadsheet visualization along with chemical structures. The third component that is kind of a must-have in all kinds of chemistry use cases is the ability to do structure searches along with filtering on any kind of data. In order to achieve that, we have included a JChem microservices component along with Marvin as a drawing tool, so that you can define your queries and you can run your structure queries in connection with all the other native Tableau filters that is provided by default in Tableau. In case you want to hear a lot more details on how these extensions work, please join our other presentation that is focused on the use and demonstration of these specific extensions. Besides the chemistry extensions themselves, we started to investigate how the visualizations can be tailored to a specific problem. So let's say the previous example about selectivity, how a dashboard can be created that is focused on the topic of selectivity itself visually, instead of providing simply a spreadsheet of numbers and structures. Also, we tried to experiment with some clickable prototypes in order to test with the users what their expectations are. Let's say in the context of R group decomposition and how that is supposed to work in the typical visual analytics approach. Not to mention the expansion towards biologics, how sequences, sequence similarities could be treated in a visual way in those dashboards. So all in all, building those extensions and then building up further and further visualizations to experiment with on the extensions was one of the main focuses for the past 1 year. So where to expand after we're familiar with the data analysis and visualization? You've already heard a presentation on the Design Hub and how it improves your experience when designing new compounds. Design Hub was one of the components that was mentioned at the very beginning on that nice graphical representation of the network of components. It is one of the components that is integrated into the whole connected solution in order to provide you the ability to extend your use cases towards the new compound design. As you've heard on the Design Hub introduction, it provides you essentially with all the possible calculated properties at a glance, so that you can see all the basic physicochemical properties, maybe some nice visualizations, radar plots, comparison options for different results, but also a quick and dirty visualization of 3D and how your design molecules fit into a certain ligand pocket. These are, of course, not meant to replace your favorite docking tools. These are much more focused on a medicinal chemist perspective, where people simply want to see a quick 3D alignment, whether that contradicts with what we know about existing ligands in the pocket or not, and something that can be used for a quick filter before even completing a docking experiment. Besides the property calculations, there are some smarter plugins like the ones that you see here, which present you an option to suggest you modifications on the newly designed compounds trained on Campbell data with the result of a more than 100, almost 200 different transformations regarding the hERG optimization. Besides the computational chemistry aspects, there are a bunch of plugins that deal with simple database lookups against, let's say, the Anamin database, or as you see here, a quick check on the legal compliance aspects of the newly designed compounds. So, how to move forward if we covered everything related to analysis and compound design, and everything is a strong word here. Well, there's still a gap that's missing in order to properly cover a research workflows, and that is the topic of being able to connect with lab workflows. So it's nice to deal with small molecule registration, it's nice to deal with assay data capture, compliance checking, and design and hypothesis management. But at the same time, people have the needs of managing samples in the lab, capturing experiments quickly, exchanging data with each other, and possibly integrating instruments or other applications in that lab. And it's not only that they have these needs, but they have these needs in connection with the components that we demonstrated. So recently we've been working quite heavily on connecting all these needs from the labs to the components that we already have. And these are the next steps that we are trying to focus on in the very near future. As a summary, I'd like to mention here that first of all, we've learned during the past 1 year how important the flexible data visualization and analysis is in such a integrated solution for the end users. Also, we started expanding our capabilities towards design workflows. And as I said on the very last slide, we have planned and we have started working on the support of different laboratory workflows. With that, I'd like to wrap up my presentation, and I'd like to invite you to try our demo environment, and also feel free to ask any questions from me either in Q&A or through email. Thank you.